In this short presentation, I will explain how we intend to employ the myeloid cell therapy platform technology in treatment of Alzheimer's disease, a neurodegenerative disorder leading to loss of cognitive function and most commonly associated with aging. In order to understand how myeloid cell therapy might work in this setting, we need to first appreciate what happens in the normal brain during the aging process. The normal CNS will be comprised of neurons, which themselves comprise of a long axon and a cell body, and these cells transmit electrical impulses. These neurons will be cohabiting the brain with cells such as microglia, the so-called macrophages of the brain. A normal interaction of neurons and microglia would be through the normal homeostatic regulation of brain integrity, for example, microglia taking care of materials budding off from neurons. Many of these processes in themselves are not usually pathogenic, and in the brains of healthy people in older ages you will find some accumulation of a protein called amyloid, or phosphorylation, which is a chemical modification, of a protein called tau. So a small degree of these is perfectly normal and non-pathogenic. However, the picture is somewhat different in conditions such as Alzheimer's disease. In a pathological setting, the same type of processes occur, but to a much higher degree. There will be more stress, inducible inflammation that will cause an activation of the microglial cells, and this will result in larger accumulations of amyloid and more extensive phosphorylation of tau within neurons. Leakage of amyloid from neurons will eventually lead to amyloid plaque formation. This will lead to loss of nerve cell function, resulting in cognitive impairment that gives the symptoms of the disease. So what differs from healthy people and individuals with Alzheimer's disease is that both hallmark features, the amyloid accumulation and the tau phosphorylation, occur at the same time and to a high degree. On this slide you see a representation of the pathological features of a late-stage disease. On the left you will see the black color that depicts extensive tau phosphorylation in the cell body of a neuron. In the middle is a large amyloid plaque, and these two features will lead to loss of nerve cell function. There will also be an associated inflammatory response represented by activated microglia and astrocytes. So the idea of this presentation so far has been to set the scene, to understand how Alzheimer's disease pathology occurs, and now let's see what we can do about it. We believe that one potential mode of therapy would be the adoptive transfer of anti-inflammatory macrophages into the brains of individuals with Alzheimer's disease, which we believe will stop or ameliorate the destructive process. We believe that an anti-inflammatory state of the myeloid cells is the correct functional phenotype. That means having the correct properties to have these proposed beneficial effects, as we believe Alzheimer's disease to be a pro-inflammatory disease that leads to cognitive decline, with phosphorylation of tau as a key feature of this process. In this slide you will see a cartoon depicting how we propose our therapy would work. Through injection of anti-inflammatory activated macrophages, preferably derived from the patient's own blood, then if they access the sites of pathology in the CNS, then they will be able to stop the tau phosphorylation and amyloid aggregation. We have demonstrated that macrophages activated in this way are better able to eat up amyloid, and this is another reason that we believe our therapy might also be effective in Alzheimer's disease that is already well established. The anti-inflammatory state will also reduce the pre-existing pro-inflammatory state of the activated microglia and astrocytes in the diseased brain. Exactly which or how many of these processes will be affected remains to be seen, but that is why we conduct a research project. So that was a 5-minute summary of this research project. We hope that you find it as interesting to listen to as we think it is to conduct. We hope that you will follow our progress by checking this website in the future.